Okay, everybody. I think we're ready to get started this afternoon. My name is Guy Lasella, and I'm the director of the Prompt Institute. Does anybody in here, are there children getting prompt therapy? We do have a couple. Great. So for those of you that don't know, prompt is a motor-based approach. We're best known for using our fingers on the articulators to help produce speech. It's a motor-based approach, but the beauty of it is in the packet that you got, you'll see a little flyer. You'll see 4 little circles. Prompt therapy is one of the few speech interventions to incorporate social emotional domain and cognitive linguistic domains, as well as that physical sensory piece we talked about. We're a nonprofit organization. We've trained about 60,000 SLPs around the world. A prompt is delivered in 100 plus countries, and we teach now in about 20 different languages. I will be around for the next 2 days if any of you would like to learn more about prompt and how the therapy works, but right now I'd like to introduce Yuri, who I believe needs no introduction to anybody here. So Yuri, you can come on up.
All right. So this time we're going to do 2 talks. So the one today is a bit more general, and I hope to cover a lot of the basics, but I wanted to put a little bit more emphasis on sort of new information that we have gathered in the last few years, including the last 2 years. Now, the last part of it will be important to be attentive and pay extra attention when we're talking about genetics, because it will be a good way to start the next talk. I'm gonna switch my slides with my left hand here. Okay, let's see. So as far as the agenda, we have 5 topics here that I wanted to cover. We're going to start with frequency, as far as how common is SAS. This is a question I get often, as far as how common really it is. Then we're going to talk about the function of the SATB2 gene, kind of what we are still in the process of learning, what else have we learned. Then we're going to talk about the clinical part, which is what I do the most, the clinical features and problems, medical, behavioral, that kind of stuff. And then we're going to talk a little bit about management, what we are currently doing. And then the last part, again, that 5th point is the one that is a little bit more in-depth and harder maybe to follow. We're going to talk about different types of genetic changes and how the change in the gene affects the protein and how it leads to SAS kind of thing. So if you're going to nap, it's probably better if you nap between 1 and 4 and wake up, so a power nap really, and pay attention for number 5. All right, so the frequency. So there's a couple of ways that we have been able to estimate how common SAS is. So the first part, back in 2019 or 18, we did a study, and if you remember, many of you guys are familiar with exome sequencing and whatnot. So there's a few labs in the United States that do it. One of the most common places that we use is called GeneDx. So they do a lot of genetic testing. So I partner with them, and then we query their database, like, hey, out of the thousands of samples that you get, how many SAS diagnoses you make, kind of thing. So that was a simple question. And from that, we were able to tell that it's really a fraction, so 0.25 to 0.3. So 3 out of 1,000 samples that they were getting for developmental delay, that they didn't have a genetic explanation. 3 out of 1,000, they were having this gene being found as the cause of the problem. So back then, we were kind of estimating, well, what is the frequency in the general population of developmental delay or intellectual disability roughly I put 1%, but it's probably higher than that, times the probability that those individuals will have changes in this gene, which is 0.3%. And that roughly gave us a 1 in 3,300. That was, again, back in 2019. I think this is when we published this. So more recently, and you see a reference at the bottom from this year, so there's been a couple of studies that have looked at that in greater detail, like what is the real frequency, not just for SAS, but for all type of genes that cause neurodevelopmental problems and whatnot. And really the summary from these two papers from 2020 and 2024, SATB2, it is roughly about 1 in 20,000 to 1 in 30,000. That is in the general population, not just individuals that have delays or whatnot, but 1 in 20 to 30,000 is really the estimates. We were not too far off from these two papers. And if you look at, okay, let's look at what are the most common genes that are mutated when you have somebody with delays. What are the most common causes of delay, intellectual disability in the general population? Regardless of the study that you use, you're going to find the SATB2 is in the top 20. So it's a much more common gene than we think. Allison was telling us the number. The Facebook group has 750 individuals. I know, of a few hundred myself. So in summary, it is much more common than we all think. We just don't see them as much, either because the testing is not being done, or they are just not reaching out to the foundation or myself or whatnot. Okay, so the second part is, what does the gene do? The cartoon at the top, that is really the rendering of the protein, what it's supposed to look like, the SATB2 protein is supposed to look like. So for the longest time, we have known the function of the gene has to do with three main systems, brain, bone, and craniofacial. When we talk about craniofacial, that's the jaw and the teeth and the palate. So we have known that for a long time. That's just based on mouse data and whatnot. So there's a couple of things that I wanted to point out here, and this is from 2018. We know that the SATB2 gene has a role before birth, that's the prenatal role, and it has a role after birth, so there's a function throughout the lifespan, which is important when we talk about treatments and whatnot. So before birth, we know that the SATB2 gene is, and you will see in the next few slides, it's a master regulator of many other genes that have different functions. So in the prenatal setting, so before birth, it has to do with neurons, which is brain cells formation and growth. And postnatal, so after birth, it has to do at that point more with how the signal is transmitted between neurons, and plasticity, which is our ability to sort of adapt to different environments and whatnot, cognition, memory, and whatnot. So it's a function that is across the lifespan kind of thing. So this is from the last couple years, which is really interesting. As I was saying, the SATB2 gene is a master regulator of many other genes. Our body has about 20,000 genes. This gene alone, the SATB2 gene, we have found that regulates over 1,000 other genes. And of course, they all have different functions and whatnot. About 170 of those genes that it regulates have to do with brain development, which is a really high amount of genes. And then this is from a recent paper from 2024. You see at the bottom wall it all. What they found is that the loss of SATB2 is implicated. It is that many genes that it affects that they estimated that about half of the genes control intelligence. So intelligence for all humans is determined by a bunch of genes. Okay, SATB2 alone controls nearly half of them. So you can imagine how important this gene when it comes to brain function, brain development, memory, intelligence, cognition, speech, all that. So from mouse data, we know that, and that has been studied before, they've looked at the mouse. If you change the gene, the SATB2 gene in mouse, what does the mouse do when it comes to their behavior, their pattern? And you recognize many of these behavior patterns are also present in humans as well. So the mouse will be hyperactive, hyperimpulsive, abnormal social behavior, impaired spatial learning and memory. So a lot of the things that happen in mouse we correlate with humans. That is well known. More recently, and this is from a paper that we just published with Dr. Fish, we have known that the SATB2 gene has a role with the jaw, the palate, the teeth. But also in this paper, we were highlighting that it has a role with the chewing muscles, so the temporalis muscles, those are the names of those muscles. And actually, the tongue in the mouse forms very differently too when you change that gene. So when it comes to speech and when it comes to swallowing, feeding, then you can start seeing why that is. So on the left side of the screen, that is the kind of an X-ray scan from the side. So it's a side view, and you see what it's supposed to look like in terms of the mouth and the tongue, just to orient you. That's not what I intended to do. So the bottom, here's the jaw, here's the normal tongue. And then you see on the right side of the picture, and I put an arrow there, that is where the tongue is supposed to be. And you see how different it is, just uneven and small. And you see that the jaw is also different. So we just got that data. More recently, and this is from Dr. Gu et al., he was looking at colon, and he found that SATB2 actually plays a role in the development of colon cells, which is interesting. There's a lot of, and this is something that we've been learning over time. So when we started doing SAS, we didn't know about all these things that we're now learning. So is this part of why maybe there's a higher issue with bowels and inflammatory bowel disease and a variety of symptoms related to bowel? I don't know the answer to that, but now we have a potential explanation. On the right side, this is also from our recent work with Dr. Fish as well. We also determined that SATB2 plays a role in the thyroid development. So, and there's a few kids that have had issues with thyroid that have needed, medication for low thyroid and whatnot. So again, there's a role that goes beyond the original brain, bone, and jaw, teeth, palate kind of thing. So there's more to it. All right, next, we're going to talk about the clinical part, the features. The cardinal features for SAS continue to be the same. When I came up with this acronym in 2018, I was trying to be clever, but it still applies actually to this day. So the main features continue to be severe speech delay, abnormalities of the palate, teeth anomalies, behavioral problems with or without brain and bone, and they all tend to be present before 2 years. So that's still the core features to this day. And the next few slides, I'm just going to expand on what we know and newer data about that. So this is data from the registry, and this is as of a couple of weeks ago. And what this is the frequency of problems. So more frequent on the left side, and that is the percentage of people that have the given problem. So speech delay continues to be the main problem. You see 100%. Teeth problems, dental, 90%, and you can go down the list and see what the main problems are. You're all familiar with some of this data. The bone density, which is the far right, has an asterisk because this is out of the ones that have been studied. It's a different number if your denominator, if you include all the patients versus only the ones that have had bone density scans. So we'll expand on bone in a second. This is from the registry as well. So when we have you guys fill the survey initially, we kind of simply ask you to quantify the speech and put it into any of these 4 buckets, right? So either no speech, 1 to 10 words, 10 to 50 words, or greater than 50 words. So this is a snapshot of all 291 data points into those 4 buckets. And then on the x-axis, you have the age. So while you see that a lot have very limited speech, especially in the first 5 years, you would think that that's maybe not completely abnormal, and it could be something else. We do have several individuals that have more verbal communication, and you see the age range. This to illustrate that there are some individuals that will continue to have some speech difficulties past, say, 10 years of age or whatnot. There's a couple of studies that are newer in the last couple of years. This was from the Netherlands and Belgium, and maybe some of you participated. They were looking at speech level, quantifying how common it was and how verbal versus nonverbal individuals would be. In general, they kind of validated some of the stuff that we have previously found. So they a little bit over half of them were nonverbal. And again, it's not only the speech production part of it, but also the receptive skills. They found that a lot of the kids, most of them actually had difficulties with the receptive, which is the understanding part of it. In the last three little donuts here at the bottom, you see the expressive language impairment. So again, that is verbal communication was affected universally. Apraxia, in the past, we have done a similar study a couple years prior to this one, and we found that apraxia was very common. Every time you tested for apraxia, then it was almost always present. In this study, they found it 75% of the time, give or take. And then the last part of it, feeding difficulties, again, another common problem, 80-90% of the time in this study. The second part of the acronym is teeth. We do see it almost universally. It can be baby teeth, it can be adult teeth. These are just collage of different problems. Teeth could be missing, widely spaced, big, rotated, fused. There's a broad variety, just looking on the outside. And then when you do x-rays, you also often find things that you might not able to see just on the exam on the outside. This is from our own work. A few years back, we did x-rays on 18 individuals that came to the clinic in 2018. We found x-ray changes in all of them. So I know that the technical issue is just having the kid cooperating to get x-rays. Tomorrow when we talk about recommendations, we'll expand on that. But still, universally, radiographic x-ray changes as well. The next part of the acronym is the B, the behavioral part of it. You guys, actually, many of you participated in this study, so this is from 2022. They describe in greater detail the profile of behavioral abnormalities that is common for SAS, and they found things that you all know, but they were quantifying. So injury, behavior, physical aggression, and a high frequency of autism and autistic tendencies. The next part of the acronym is the brain, so the seizures part of it. We have done this study a few years back. We know that seizures happen about 20% of the time, but we do also know that EEG abnormalities are more common. So not everybody that has an EEG abnormality has seizures. So twice as many kids and adults have problems with the EEG, even if they are not being labeled seizures. In this study from 2020 that we did, we looked at the data from EEGs that was available to us, and every time an EEG was done and included sleep, it was abnormal. So that's part of why we are, when we talk about recommendations tomorrow, we'll talk about that too. In this study, we found that seizures, the average age of onset was about 3. We saw a broad range, 4 months to 6 years. I'll tell you that I know of older individuals than age 6 that have started having seizures. So I never say never kind of thing. When they ask me, okay, is my child seizure free for good? If it's 10, then I would say it's probably less likely and it's going to be a lifetime risk, but it goes lower as the age advances, kind of thing. The next part of the acronym is BONE. We have known this for several years. In 2017, we had done the first study. We know that broken bones, scoliosis, bowing of the limbs, those things were present. The newer data comes from two sources. One part is from the group in France. What they did x-rays on a bunch of individuals with SAS, on everybody, and see what the common findings were. So I'm just pointing out a couple of the things that they found. So they, for example, when they look at the skull, they found what we call cranial impressions. So cranial impressions are those little like bumpies that you see on the skull. They really don't mean much in terms of is it bad or whatnot. It's just another finding that we're seeing in SAS. And they, interestingly, when they did the x-rays, they did it on everybody. And about a third of the patients that had the x-rays, they found what we call compression fractures, which it could be kind of a sneaky broken bone to detect, and a lot of times you need the x-ray to see it, or otherwise you might not know, but it's still there. So that was from the French group. Then last year we published our data, which it was a bit larger data set. So combining the French group and our data, this is kind of the summary. So we found that about a third of individuals with SAS had a previous broken bone, no matter the cause or how it happened. We found that when you do a bone density, you find it to be really in the low range, so what we call osteopenia or osteoporosis, in about 25% of the time. Now, there was another 25% that had it low, but not super low, if that makes sense. So ultimately, about half of the time, it was abnormal. It was just a matter of how abnormal it was in most cases. And then we also have been collecting blood to do kind of bone markers for a while too. And what we found is that there's a chemical in the blood. It's called alkaline phosphatase, right? It is produced by a few parts of our body. Bone being one, it's produced by the gut, it's produced by the liver. So we found that if you do alkaline phosphatase, it will be abnormally high in 62% of the time. And what we did is, okay, is it really coming from bone? And we found that, yes, it is coming from bone. So it kind of makes sense. The bone, in general, in SAS is just constantly being remodeled, probably at a higher pace than an individual without SAS. And the alkaline phosphatase is just telling us it's going through remodeling. So it doesn't really mean, and we tried to see if it correlated with a low bone density or broken bones and whatnot. It doesn't. It just tells you that there's a really high activity in the bone happening. This is also from our recent work with Dr. Fish. We knew that the jaw in individuals with SAS appear different many times. We presented in this work images, which is not really done routinely. So when you look at the CAT scan, which is the left and the middle, we saw a small jaw. We saw that there's no symmetry also, like left and right side of the jaw look different. And the farther right is just a simple x-ray. You can appreciate how small the chin is in proportion to the upper teeth that protrude out, kind of thing. That's on the x-rays. If you look at chins of individuals with SAS, it's all over the place. Some of them are a little pointier than others. You see kind of a triangle or slightly more kind of sticking out, like the lower right side. Okay, so in general, SAS is a very hard diagnosis to make unless you have some extra clues, which is what Allison was sharing with us earlier today. So I've been trying to kind of put it in a spectrum of when do you see this problem kind of thing. So this is what I came up with. So cleft palate, which was Allison's story too this morning. Cleft palate, of course, will be present at birth. It's an easy and day one of life clue. The low muscle tone is probably what follows in infancy and the first few months, along with the feeding difficulties. Then, you get to the 1 year and 1 to 2 years, really, is probably when you will start seeing developmental delay, especially in speech. Obviously, will be the more prevalent thing when they don't start speaking or similar. Growth in general. So if a child has a cleft palate with feeding difficulties, of course, the growth might be affected sooner than that. But for the majority, that's not the story. So we see a slow growth, but that tends to be childhood and later. The teeth problems, of course, you would need to be old enough to have all the teeth come in. And if you're going to have problems with adult teeth, you might not see it until you start losing teeth, or you're not losing them, and that is the abnormality. So that's really more of a childhood problem. The behavioral problems, I'm putting here childhood along with the mental health and sleep. And then seizures, I mentioned the average age was 3 years, but of course you can see it before or after. And the bone anomalies in general tend to be, again, kind of a childhood problem. So that's roughly my guess of when things happen, kind of thing. Okay, next, I'm gonna briefly talk about management. As you all know, and I can understand how frustrating some of these things are when you're talking to your provider. So there's no specific treatment for SAS, and really what we concentrate on is prevention of problems and kind of managing the symptoms as they happen, which is, again, often for many of these neurodevelopmental syndromes, and we're trying to change that. I'm going to just expand on a couple of things when it comes to treatment, and tomorrow we'll talk a little bit more as well. So speech, our team has evaluated now over 100 individuals with SAS since we started this, so we have good experience when it comes to all this stuff. Speech-wise, our speech team has put letters and recommendations. We don't go any less than 90 to 120 minutes, and I understand that, a lot of times it's like you don't want to only get that if you have it on a letter, so we always try to advocate for no less than that, because many times they will try to give you less than that. As we heard AAC today, we recommend considering AAC from early stages along with feeding therapy. From the behavioral perspective, and I put my disclaimer here at the bottom, always consult with a mental health provider. These are things that I don't even manage myself, to be honest. These are many times complicated situations that need multiple people and treatments. David told us earlier about behavioral management plans, so often that is a good starting point. I often get asked, like, is there one medication that works better than others when it comes to behavior and sleep and whatnot? And the answer is no. I am happy to tell you what has been used. And in every instance, there might be some that work for a child but did not work for the other one. I always encourage to get mental health evaluation and an actual diagnosis, whatever it is, ADHD, OCD, anxiety. That's much easier to find a treatment for than unspecified behavioral management kind of thing. So for hyperactivity, impulsivity, you will hear us talking about clonidine, guanfacine, different stimulants for ADHD purposes. Anxiety, our team uses Lexapro a fair amount, and this is, again, a child psychiatrist that manages the SAS individuals in our team. Sleep, there's kind of a progression of treatments. Melatonin is usually a starting point, along with sleep hygiene and whatnot. From there, we often go to clonidine. From there, there's multiple options. There's some individuals that require trazodone, for example, which is a stronger medication. For aggression, there's, again, a variety of things. You'll hear us talking about Abilify, Risperdal. any of these things will have pros and cons, and it's always hard to balance benefit versus side effects kind of thing. All right, so when it comes to bone, we measure vitamin D, calcium, try to optimize the bone health, the things that are easy. If the child is low in vitamin D, then we just give more vitamin D, kind of thing. There's a group of medications called bisphosphonates. We use those sometimes. That is a medication that is often used in adults with osteoporosis and postmenopausal sort of bone fracture related. Is a very strong medication. We save it for situations where it's really needed. It's been used in kids for decades now, so I've used it several times. We try to save it for situations where we have documented low bone density, and especially if the individual has had broken bones. So that those will be the requirements to need it. They do respond fairly well to it. The bones do get stronger as far as we can tell from repeat bone density. Seizures, again, there's no one seizure medicine that I would say works better than others. We know that often more than one seizure medicine is needed. From our study in 2020, we asked all the families that had seizures, hey, what was your child on? What was your experience? What was the first treatment? And then what was that switched to? So we know Keppra, which is a very common medication for seizure, was the first, often the first treatment used. The problem with Keppra is that it has a few side effects, including behavioral issues. So a lot of times was making behavioral issues worse. So many of those individuals resulted being changed to a different type of medication, either Lamictal, Trileptal, Depakote, Omfi. So you name it. There's multiple medications that can be used. I know maybe one or two that have tried a ketogenic diet, and that was in very severe seizure setting with mixed results. So it's not anything that we do on a regular basis. Okay, and we're down to the last part, which is the genetic aspects. So if you, I'm going to walk you through all these slides slowly. So we know, as far as SAS, we know as far as how likely is it to happen again, and why did it happen. So most of the time, you will hear us talking about de novo, right? So de novo means brand new. Almost always, 99% of the time, it is brand new. So if you go do testing in blood in the parents, you're not going to find a genetic change. That's what de novo means. So, however, with that said, there's a 1% chance that it could happen again. And we have in our registry, and we know of several families that have had more than one person affected in the family. So there's different ways to know that. So of course, if you do the testing in the parents, sometimes you will find that a parent could have the same change. It's just at such a low level that it's not enough to cause problems. So sometimes we do see that. And then there's times that you do the testing in the parents, you don't find it. That doesn't mean that the risk will be zero, but it's lower maybe than if the parent had that change detected. So those are conversations that we had with families all the time. We don't say never. We just say, most likely it will not happen again. There's a 1% chance. I know of a family, one of the parents had it in blood at such a low level that wasn't causing problems, but enough to have 4 kids affected with SAS. So that family I follow as well. Okay, so really basic genetics here. So when you look at, if you remember, chromosomes is where all our genes are packed. We have 22 pairs. We have half coming from mom, half coming from dad. Chromosome 2 is where the problem is, right? If you think of it as chromosome 2 being the city that you live in, then you have to zoom in and you see that highlighted area right there in red. then you kind of zoom in and you see this number at the bottom, the 180 million to 215 million. That is roughly kind of a zip code, if just to put a comparison. So the SATB2 gene, if you go back and look at your genetic report, it's going to tell you the address, exactly where the change is, right? Roughly, it will be in the 200 million mark, give or take. So sometimes it will be 199 million, sometimes it will be 200 and change million. It depends on how the test was done. But what we do know is that the SATB2 gene around it has a bunch of other genes that have different functions and could cause other problems. So again, just to try to keep it so it could be individualized to each one of you. So if your child has a large deletion, we call it, so it's a chunk of chromosome 2 missing, so it will be a SATB2 gene with other genes around it. So depending on what other genes are missing, then we might be able to tailor specifically what we will tell you. And tomorrow when we talk about guidelines, then we'll talk a little bit more about that. Okay, if you continue to zoom in into that gene, then you get this kind of image. On the top is the actual SATB2 gene, and you see a little boxes from 1 to 12. Those boxes are called exons, and the exons, we think, we as in all scientists and medical community, is where the majority of genetic diseases are located. So most of the information that you need to form proteins is located in the exons. So most of the time, if you're going to have a genetic change, it's going to be in the exons, right? So from the gene, you need those are the instructions. Your body will produce a protein, which in this case is a SATB2 protein, which you see in the middle of the graphic. And you see the number goes from 1 to 733. So that is the amount of building blocks the SATB2 protein has. Also, if you go back to your genetic testing result, it will tell you C dot blah, 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 which is the top part, and then it will tell you P dot blah, 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 which is the second part. So it will tell you exactly where the change is. You also see, I'm going to show you this middle image several times today and tomorrow, so you'll be familiar with it. You see the little boxes, the purple ULD, CUTL, cut 1, cut 2, and the HOX. So those are, we think, areas of the gene, of the protein that have a more prominent role. They have very specific functions. So, and that would be important because it matters where your gene change is located. And at the very bottom, again, that's the same cartoon I used previously. So the middle one and the bottom one are the protein. It's just the middle one is the pretty version of it. No protein in our body looks like a straight line, of course, so they look like the mess at the bottom. So that's just for easier to see image kind of thing. All right, so if you look at the genetic code, so again, if you zoom in into a chromosome, you get to a double strand, which we're all familiar with. We all have a sequence of letters, two-stranded, right? So, and our human code has the 4 letters TACG with a counterpart to form the double strand. So all that because when I talk about the different types of genetic changes, then that kind of makes sense. So on this image, we have what we call the wild type. Wild type means normal for our purposes. You have the double-stranded DNA, you see the letters. Those letters are used for the instructions, which is the middle, the mRNA, which is called messenger RNA. Those are actually the instructions that are filtered, and then that is the blueprint used for our body to produce the protein, right? So the different genetic changes that could happen for any gene, for that matter, but for SATB2 purposes, this will be relevant in a second. So you see what happened here. The first part, we call it a missense change. In this case, it was supposed to be a G. Try to zoom in here. And now we have an A. Okay, so that letter is not supposed to be there. That means the instructions now are different. And a missense change, what happens, and you see at the bottom, there's a building block that now is a different building block. But otherwise, the protein is all the same. So it's just one building block that is different. The other 732, because I told you it was 733 building blocks, is the same. So just one little building block that is different. So that is called missense mutations. The second type is a nonsense mutation. For a nonsense, you're still replacing a letter for a letter. However, when your body reads that letter, it's going to tell the body to stop building the protein. So there's a very specific kind of letter code that we follow. What happens here at the bottom, you see now the protein was fine, and once it encounters this new instruction, it stops. You see that little star mark at the bottom. So it's a much shorter protein than it needs to be. Another type of mutation is what we call frame shift. So the frame shift, instead of having a letter that is different, now you have either a letter, just one letter or two letters missing, or one or two letters that are extra. Either way, it will mess up how the body is reading it, because the body is trying to read a sequence, and now you have letters that are not supposed to be there. So when that happens, the body is going to produce the protein fine until it finds the messed up instruction, and it will continue to build that protein. But now it's a completely different instruction, right? So it's going to continue to build that protein, and at some point it's going to stop. It's just not normal anymore. And then the last type of mutations are called splicing. I mentioned really briefly what an exon was. So splicing mutation is really at the end of that exon. For our purposes, it should behave similarly to a frame shift. So it's a different instruction. It's going to result in a protein that is not normal either way. Okay, so this graphic, in addition to the registry, I try to keep an eye on what others are publishing, and I keep a mutation database, and that this is what this figure means. So out of all the individuals that have been diagnosed with SAS that I know the mutation, and this is also as of the last 2 weeks, you see that all the things that I just told you that are represented here. So you have in green, you have your deletions, again, a chunk missing, different sizes, of course. In purple, you have what I call a small deletion. So that's only missing a part of SATB2, not SATB2 and something else. In red, you have the missense changes. So about 30% of the individuals have missense changes. And then in blue, we have nonsense, frame shifts, and splice sites that we call truncating. So almost half of the individuals have that type of mutation. With that information, then you can put it in a map, which is in a protein map, which is what I did here. You see that there's some mutations, some changes in the gene that are recurrent, so more than one individual will have it. The most common mutation that we have, you see it in blue right in the middle, is called arginine 389 cysteine. It's a missense change. 33 people have exactly the same change. So in blue, we have missense. In orange, we have nonsense. In red, we have frame shifts. But the bottom line is that you see it across all over the protein, right? And the boxes with the little dots, pins, those correspond to the same figure that I've shown you now a couple of times at the bottom. So the little, I'm going to try to point here, the little orange box here corresponds to the purple, this blue one corresponds to the brown, etc. So we can map them. And that is how a lot of times we can tell if it's going to mean anything or not. So for example, now you can see this is kind of a density map, right? So where are all the mutations located? Is the gene tolerant to change in that specific spot or not? Or is it intolerant? And that is this heat map that it generates here. So the lower it is, did not mean to do that. The lower it is, or the redder it is less tolerant to change. The yellower or bluer it is, the more tolerant it is. That means that if you have a change where the blue is more likely that you won't have SAS, versus if you have a change where the red is, it's more likely that you will have SAS because it does not tolerate change in that area. Okay. We're going to conclude here with a couple things. So, as you know, SAS is a dominant genetic condition. That means that you all have, myself included, we all have 2 copies of every gene, right? So for a dominant condition, if you have a problem with just one of the 2, that's enough to cause problems, okay? So that's what dominant means. Now, the difference is, how does the one get affected? How does one copy that doesn't work, why doesn't it work? So there's 3 different ways that can happen. So we call haploinsufficiency, dominant negative, and gain of function effects. So I'm going to walk you through that, and that will be relevant as we talk about treatments and our next talk. And tomorrow I'll be revisiting some of this stuff. So in haploinsufficiency means half of it works, half of it doesn't work. So in haploinsufficiency, the second option here, so if you have a nonsense or a frame shift, the protein didn't form well, is either shorter or messed up somehow, our body will try to get rid of it, and then in the end, you end with one copy that is normal, and then the other copy that produces a protein that is not going to work, and our body is going to get rid of it, so no protein. So half yes, half no, so that is haploinsufficiency. There's a different model for dominant disorders, and this is part of what we're trying to understand, that would be you still have your normal, but then the second part of it is that copy that has the SATB2 mutation is still able to produce something, and it's going to interfere with the normal copy. So we call that dominant negative effect. And then the last one, what if that change in the gene, instead of having less SATB2 protein form, produce more SATB2 protein? And that also will be critical to know, because if I'm talking about treatments and I want to replace SATB2, well, I don't want to give SATB2 to somebody that is producing too much SATB2. That would be detrimental. So when it comes to SATB2, we know this group, the nonsense, the frame shift, and the splice mutations, those are going to be relatively similar because they all result in a protein that doesn't work. But for missense changes, that's the part that we don't know. So tomorrow, I'm going to be talking about that. And with that, that was my last slide. Of course, you all have my email. I'll be here today and tomorrow and Saturday, part of it maybe. So you can ask me questions at any time if you don't get to ask me questions right now. But that's what I have.
Hello. So I thought perhaps what could be a nice add when you were talking about the medications, I think a lot of us have seen you mentioned hyperactivity, anxiety, and sleep. You offered some medications. We've also seen and I thought you might want to include is those intermittent moments when our children need to have a DEXA scan or need to be some sort of intermittent sedation, really without having to go to that whole anesthesia. I thought perhaps you might want to consider including lorazepam. I have seen several friends, and I have it, and we haven't used it yet, but Ray is awful in terms of a dental examination. So those type of things I thought perhaps you might want to include adding or discussing if you're seeing that. And then another one I wanted to mention was, or a question I had really was when you talked about the bone density. Do females, specifically with the menstrual cycle, are you seeing in this study that there might be more impact on a female because of the estrogen, testosterone stuff, I'll learn soon enough, that happens during a female's puberty time? Yeah. bone density, seizures, etc., around females.
I don't know. No. Okay, so thank you for the suggestion for sort of the temporary sedation and acute management of those specific moments. And honestly, we haven't done a whole lot of that. I guess we still rely on other things that are not medical, like, to help ease some of these things. So I appreciate the recommendation. Thank you. For bone density, we have not seen any gender differences. So we see it males and females, and we see it starting as early. I've seen lower bone density in infants too. So it's starting from infancy to later. So and tomorrow we'll talk about recommendations. We typically would say 3 years is really when most people will feel comfortable reading a DEXA anyway in a child. But we have not seen differences based on sex.
This question is from the online, and it comes up often. So what is the likelihood of an unaffected sibling having a child with SATB2? Affected child is de novo.
Okay, so that is a relatively easy answer. So for an unaffected sibling, it will be 0%. Or I wouldn't say 0, sorry. General population risk is probably a better answer. So it is not going to be more likely than somebody that doesn't have the family history for an unaffected sibling. That is relatively easy. Even if the parents had proven to have some low level in the blood somehow, and then the sibling, unaffected sibling gets tested and is 0, then the answer is you're not more likely than somebody else in the general population. So that is not bound to happen again.
I know that we could all stand here and ask questions all day. We are going to take a quick break. We're going to the next panel. Our speaker will be joining us virtually. So get a quick break, come back. We'll have more time for Q&A after that session and more time tomorrow with Dr. Zarate.